back to the channel. Let me begin with a simple question. How life on Earth learned to use sunlight? Today, plants dominate our landscapes, and photosynthesis is often associated with green forests and flowering plants. But the fascinating truth is that plants were not the first organisms to capture sunlight. Billions of years before the first plant appeared, tiny microorganisms had already evolved the remarkable ability to harvest light and convert it into usable energy. This innovation transformed Earth's history forever, ultimately leading to the oxygen-rich atmosphere that supports complex life today. In this video, we'll explore the evolution of phototrophs, understand how photosynthesis began, and discover how microscopic organisms changed our planet forever. To understand the evolution of phototrophy, we first need to understand the difference between two major nutritional strategies: chemotrophy and phototrophy. Chemotrophs obtain their energy from chemical compounds, whereas phototrophs obtain energy from sunlight. Scientists believe that the earliest life forms probably lived around hydrothermal vents on the ocean floor, where they relied on chemicals released from the Earth's interior. These hydrothermal vents provided a constant supply of reduced compounds, such as hydrogen and hydrogen sulfide, that served as energy sources for primitive microorganisms. Although some researchers have suggested that phototrophy may have evolved very early, perhaps even before chemotrophy, the commonly accepted evolutionary scenario assumes that chemotrophs appeared first and that phototrophs evolved later. One important misconception is that the first phototrophs were plants, or even cyanobacteria. In reality, the earliest phototrophs were simple microbes. Fossil evidence, such as ancient stromatolites, suggests that microbial communities capable of photosynthesis existed billions of years ago. However, the earliest phototrophs were probably not oxygen-producing organisms. Instead, they belonged to a completely different category known as anoxygenic phototrophs. This brings us to one of the most important concepts in the evolution of photosynthesis: the difference between oxygenic and anoxygenic photosynthesis. Oxygenic phototrophs, such as modern plants and cyanobacteria, use sunlight to split water molecules. During this process, they release molecular oxygen as a byproduct. This oxygen eventually accumulated in Earth's atmosphere and made aerobic life possible. Anoxygenic phototrophs, on the other hand, do not split water. Instead, they use compounds such as hydrogen sulfide, molecular hydrogen, or ferrous iron as electron donors. Rather than producing oxygen, they produce compounds such as sulfate. Because they did not require water splitting, scientists believe that anoxygenic photosynthesis evolved long before oxygenic photosynthesis. Why did phototrophy evolve in the first place? Imagine the earliest chemotrophic organisms living around hydrothermal vents deep beneath the ocean. Their survival depended entirely on chemicals emerging from these vents. As life attempted to spread into new environments, organisms needed an alternative source of energy. Sunlight provided exactly that opportunity. Instead of depending only on hydrothermal chemicals, organisms began to exploit the enormous and virtually unlimited energy available from the sun. This adaptation allowed life to expand far beyond hydrothermal vent ecosystems. One of the earliest light harvesting systems may have involved a pigment called bacterial rhodopsin. Unlike chlorophyll, rhodopsin does not perform photosynthesis in the modern sense. Instead, it captures light energy and uses it to pump protons across the cell membrane. This creates a proton gradient, and the flow of protons back into the cell through the enzyme. ATP synthase generates ATP, the universal energy currency of living cells. In nutrient-poor environments where chemical energy sources became scarce, this simple light-driven system may have provided microorganisms with an important backup source of energy. As evolution continued, more efficient pigments appeared. Bacterial rhodopsin was eventually complemented or replaced by bacterial chlorophyll, which could absorb a wider range of light wavelengths, including infrared radiation. This improvement allowed early microorganisms to capture more energy from sunlight. Interestingly, the ability to absorb infrared radiation may also have enabled these microbes to detect nearby hydrothermal vents because hot objects emit infrared radiation. However, despite using sunlight as an energy source, these organisms still depended on electron donors such as hydrogen and hydrogen sulfide that were available primarily around hydrothermal vents. Consequently, they remained closely associated with hydrothermal environments. Another major evolutionary innovation was the development of the photosynthetic reaction center and the electron transport chain. When light strikes a pigment molecule such as bacteriochlorophyll, electrons become excited and move through a series of electron carriers embedded within the cell membrane. This chain of electron transfer reactions generates another proton gradient, which ATP synthase uses to produce ATP. This mechanism remains one of the most fundamental energy-producing processes in biology today. Scientists have long debated which bacterial group first evolved photosynthesis. Since no known archaea perform photosynthesis, it is widely accepted that photosynthesis originated within the bacterial domain. Several groups have been proposed as the earliest phototrophs, including purple bacteria, green non-sulfur bacteria, heliobacteria, and green sulfur bacteria. Unfortunately, determining the exact evolutionary sequence has proven extremely difficult because of extensive horizontal gene transfer. Unlike animals and plants, bacteria frequently exchange genes with unrelated species. As photosynthetic genes spread across different bacterial groups, reconstructing the true evolutionary history became remarkably complicated. Among the earliest candidates are the purple bacteria. These organisms use bacteriochlorophyll to absorb near-infrared light and can be divided into purple sulfur and purple non-sulfur bacteria. Purple non-sulfur bacteria often use molecular hydrogen as an electron donor, while purple sulfur bacteria use hydrogen sulfide. Since molecular hydrogen was abundant near hydrothermal vents, these bacteria may represent some of the earliest phototrophic organisms on Earth. Green sulfur bacteria appear to represent a later evolutionary stage. They possessed improved reaction centers containing iron-sulfur clusters, allowing more efficient electron transport and greater energy production. These bacteria thrived in oxygen-free, sulfide-rich environments, particularly within the lower layers of microbial mats, where sunlight was still available but oxygen was absent. Microbial mats played a critical role in early evolution. These layered communities of microorganisms allowed different species to live in close association. They exchanged nutrients, protected one another from environmental stress, and created ideal conditions for horizontal gene transfer. Modern stromatolites provide living examples of these ancient microbial ecosystems and offer valuable insights into early Earth's biosphere. Life on early Earth also faced another serious challenge: intense ultraviolet radiation. Before oxygen accumulated in the atmosphere, there was virtually no ozone layer to block harmful UV rays. Early phototrophs therefore evolved several protective mechanisms. Living beneath the upper layers of microbial mats provided natural shielding. Iron-rich minerals and silica deposits also acted as primitive sunscreens by absorbing harmful ultraviolet radiation while allowing visible light to reach photosynthetic cells. In addition, many phototrophs evolved carotenoids, accessory pigments that protect cells from excessive light and reactive oxygen species while also enhancing light absorption under low-light conditions. The greatest breakthrough in the history of photosynthesis occurred with the evolution of chlorophyll-based oxygenic photosynthesis in cyanobacteria. Unlike earlier phototrophs, cyanobacteria evolved the extraordinary ability to split water molecules using sunlight. This reaction released molecular oxygen as a byproduct. Water, unlike hydrogen sulfide or molecular hydrogen, was available virtually everywhere. As a result, cyanobacteria were no longer confined to hydrothermal vent systems. They rapidly spread throughout the sunlit regions of Earth's oceans, colonizing shallow coastal waters and eventually dominating marine ecosystems. Evidence suggests that cyanobacteria probably evolved between 3.5 and 2.7 billion years ago. However, Earth's atmosphere did not become oxygen-rich immediately. The Great Oxidation Event occurred much later, approximately 2.4 billion years ago. Why the delay? The answer lies in Earth's chemistry. The oxygen produced by cyanobacteria initially reacted with enormous quantities of reduced minerals and volcanic gases present in the oceans and crust. Only after these chemical reservoirs became fully oxidized could oxygen begin accumulating in the atmosphere. The rise of oxygen dramatically changed life on Earth. Although oxygen is essential for most modern organisms, it was highly toxic to many early anaerobic microbes. As atmospheric oxygen increased, numerous anaerobic species became extinct, while others survived only in isolated oxygen-free environments. At the same time, evolution favored organisms capable of using oxygen for aerobic respiration, a far more efficient way of generating energy. This innovation ultimately paved the way for the evolution of complex multicellular organisms, including animals, plants, and eventually humans. In summary, the evolution of phototrophy represents one of the greatest milestones in biological history.